CS201 talk. Thank you everybody for being here. It is my privilege and my pleasure to introduce today's speaker, who is Mayank Mehta. He is a professor at UCLA in 3 different departments, physics and astronomy, and as you can see it says here, neurology and neurobiology. And teaches a class, I have understood, he will say more, that cuts across all of those 3, and also using techniques from computer science. He is originally from Bombay University, and later did a PhD in quantum field theory from the Indian Institute of Science in Bangalore. And then even later came and worked at MIT and later at Brown. And then for the last possibly 4 years, 3 years, he has been a professor at UCLA. And here he is. Over to you.
Thank you. Thank you. Thanks everyone for coming. There are several seats up front here for those of you who want to sit. You can stand here too. So how the mind emerges from the brain is the topic I'm going to be talking about. There's a lot of excitement about this, starting with presidential initiative in this topic. And the first thing I should declare is we don't know. We don't know how this emerges. So this is meant mostly for you young people who are going to figure out how this thing works. So mind is something that we have some idea right now. I'm talking, you're listening. Our minds are engaged with each other. We do all kinds of behaviors. We write, we breathe. These are the things about the mind. But what about the brain? And what's the relationship between the two? So let's get some basic facts together, and I encourage you to ask questions. I don't want this to be just a long thing, so just raise your hand and interrupt, and if I don't look at you, shout, because there's going to be very complicated talk. So that's the size of the brain, human brain. It's about that much, not really big. It's about 1/50th of the human body. Although it's 1/50th of the human body, 1/3 of all the known diseases, as we know so far, are brain-based. So something is weird about that part of the brain, that even though it is such a tiny fraction of the body, there are so many diseases that influence it. So why is that? Why is it that there are so many diseases there, and what does it do? So when you look slightly below, deeper inside the brain, there you find that inside the brain there are neurons. And that's one neuron, and that's another neuron. And that's how many neurons there are in the brain. About 100 billion in human brain. It's a lot of neurons. Now let's get some terminology right. The neuron has a body like all the cells in your body. It's called soma or body. That's right there, that's the body. That's about 10 micrometers. But what makes neurons special are these other processes which are going off like branch-like processes. These are called dendrites. And these can go millimeters and sometimes all the way from here to your leg, meters. And all this takes up a lot of volume. In fact, these processes are the ones that take up a lot of volume, and for good reason. You may ask, why does it have this branch-like structure? What's the point of all of that? Let's get down to that, brain function. The point of it is that each neuron actually has 10,000 connections, both at the level of input and at the level of output. And these connections are formed on these branch-like processes that you saw, called dendrites, and the output called axons. They do really interesting things. How many synapses total? That's how many synapses, quadrillion. You think that's a lot of numbers? We haven't even started yet. Because information in the brain about anything, about the physical world or behavior, those things are encoded by the pattern of activity of neural responses. It is not that one neuron represents one thing and other neuron represents coffee and third tea and so on. No, it's distributed across a lot of neurons. So it's a combinatorial code on top of all those zeros. So number of combinations are mind-boggling. On top of that, there is a division of labor while neural activity represents information, that information is not memorized or not learned by neural activity. It is memorized or learned by the pattern of synaptic strengths. The synapses or connections in the brains are not stationary. They are changing on all time scales, on the time scale of milliseconds to time scale of lifetime. So many, many orders of magnitude time scales over which synapses are constantly changing. And still your percept is the same. You still think that I'm the same person talking. And in these few sentences we went through, your synapses have changed a lot. All of them. And in fact, if you take you take various imaging pictures, you can see them wiggling around. In fact, physically, they change, they shape. Not just the electrical connectivity, they change. All of these quadrillion of them, and it is the combinatoric pattern in which these synapses are doing stuff. That's how information is learned and then remembered. So it's a tough problem. How is this coordinated? And who coordinates this in the first place? Because it's an enormous problem. So there is massively parallel and distributed coding. It is parallel because all parts of your brain are active pretty much all the time. It is distributed because every single thing you do is distributed across many, many parts. Unlike computers, digital computers that we have been using. So that's very, very quick crash course on what that stuff is. Now let's get into brain activity. Fundamental fact, whether you look at human brain or a rat brain or a mouse brain, Einstein's brain, my brain, same thing you find across most of neurons. All neurons are sitting around not at 0 volt with respect to ground. They are sitting at minus 65 millivolt, a magic number. Nothing to do with the speed limit on the road, but it's 65. Now what happens is that for some reason, if current gets injected into the neuron, typically from these synaptic connections, 10,000 of them, and those currents that get injected, if they push this membrane potential of the neuron beyond minus 45 millivolt, just a 20 millivolt difference, then the neuron literally goes nonlinear. And that's where a Nobel Prize was given to Hodgkin and Huxley about how exactly they go nonlinear, and that generates something called an action potential or a spike. And if that seems too abstract, you're going to hear and see it in 1 second. Well, a minute. So let me just describe what those things are, because it will be too fast for you to feel that. Each spike is about, it's a very brief voltage fluctuation that is about 100 millivolt, 0.1 volt. Now imagine, sounds very little, but you have 100 billion neurons, each one capable of generating a burst of 0.1 volt. And it lasts about 1 millisecond and then it's over. So how does it feel and sound? Let's do this. This might be a little loud. And I'll explain in a second what this is. So this is real time. This discovery, what's going on here, got a second Nobel Prize to Hubel and Wiesel. What's going on in the first picture is this. There's an oscilloscope, of course. And all that somebody did is to inject current into a neuron so that its membrane potential went up just 20 millivolt above. That's what happened here. And you will see now when I say this, if you look carefully, you will see the membrane voltage show that spike like underlying burst. You see that? See that trace lingering around? That's somebody injecting current by hand. A neuron is doing this. Gives us an idea how that works. And that's what this does. Now here, on the other hand, it was not a single neuron in which somebody was injecting current, but all that was going on was somebody was sitting in front of the subject and just moving a bar of light, about that big, just moving back and forth. And whenever that bar of light crossed a certain space, point in space, this neuron responded. That neuron seemed to care not for the color, not for the smell, not the reward of the experimenter, none of it. It simply cared for the orientation of a bar. And that's that neuron to respond. And that's why you don't see that subthreshold activity. There's a lot of interesting stuff there. So that's what is going on all the time in your brain. Now, first thing I should tell you is that once you start getting into the brain, you must leave introspection out. And this is a good example. Right now you heard those spikes, and I told you that 100 billion neurons in your brain are firing spikes right now. Anybody heard a spike from inside your brain? So if you did hear, talk to some doctor. Something is seriously wrong. So we cannot introspect. It's a very complicated organ, a huge amount of stuff it does. We have no conscious percept of it. It's impossible to perceive it for good reasons. And hence is this divide, that there is this mind that you and I can understand, and there is this brain which enables us to perceive the mind and interact, but the brain itself we cannot perceive, not even our own brains. So we need to think slightly differently about this, unlike thinking about the arm where you can perceive and think and so on and so forth. Now let's go to the next step of this abstraction, brain activity and learning. What happens is this, that whenever this neuron fires a spike, it activates all those 10,000 output synapses to which it is connected to. The activity of this synapses injects ions into the output neuron, which are sodium, potassium, chloride, and calcium. These are the most common ions. These are all charged. Chloride being negative, all the other three being positive. And those charged ions get into the other neuron, change that neuron's membrane, and as a result, that neuron will be either more excited or less excited. Very roughly speaking, there are two kinds of neurons in all brains of mammals. Excitatory neurons, which inject the current in other neurons in such a way that membrane becomes more positive, roughly speaking. Makes it more likely to fire a spike. And inhibitory neurons, which injects current so that other neuron kind of shuts down, kind of cancels out. Now what happens is, what makes the brain really special is that as soon as a synapse is activated, immediately that activity of that synapse changes. So this is like quantum mechanics, where the act of measurement changes the system. Same thing goes on here. As soon as you see a stimulus, such as a simple oriented bar, that stimulus is going to activate the neurons, which is going to activate those synapses, but those synaptic strength is going to change depending on the pattern of activity of neurons. That's the key thing. And we're going to look at how exactly it depends on that. And that's roughly how this thing works. That's roughly how all of us work. That little thing is how we learn anything from walking, from being babies to talking and mathematics and so on, simple to esoteric. So now that we come to that idea of emergent phenomena, the best example of emergent phenomenon that everybody gives, and perhaps the best achievement of humanity in this regard, is in thermodynamics, which was shown by a long time ago by Boltzmann, saying that things that we can feel, when I touch something, I feel a certain pressure, I feel a certain volume, certain temperature. Those things that we can feel, they arise because of the speed at which molecules are hitting each other. You can't see those molecules, you can't feel them. If you really looked at them, you won't even see them move. But huge number of them jiggling around generates this perception of pressure, volume, and temperature, and a simple equation of PV equals NKT that allows us to do all kinds of things, make cars, rockets, novel materials, all kinds of applications. And the question is, can we think of how to go, just like people figured out exactly how to go from root mean square velocity of particles to pressure, volume, temperature, can we go from this dynamics of spikes and synapses and their change to things that we understand? Because if we understand it, we have a way of intervening it. And it is important to go from bottom up rather than top down. And I'll give you an example of that. And that's the question. And several people realize this, and we are fortunate that the President of the United States realized this is important. And they have come up with this Presidential Brain Initiative, and there are 9 points that they have suggested we should do. Point number 1, first thing is necessary is to create a census of all cell types. You saw that little blurb there that there are so-called neurons, they have all kinds of shapes. Some neurons look like pyramids, some look like stars, some look like chandeliers, and so on. So how many cell types are there? Because each cell has a different function, and they still have to work together. So that's the first part of that thing. And these are the grand challenges.
If you had to guess, how many would you say?
I would say at least 100. And in fact, it's a funny thing. the predominant number of neurons in your brain, about 80%, they are all excitatory. They don't have such a huge diversity, but a minority of them, which are inhibitory, they have the largest amount of diversity. And there's an expert here, Iswan Modi, who knows all about it. But I won't say any more because I'll say wrong things. But there is a lot of diversity there. Now, once to make these maps of these neurons, now we want to develop new large-scale network recording techniques, because we agreed that neurons are active at the same time, they're active very fast, so you want to pick up the activity of lots of neurons very quickly and simultaneously, not one neuron at a time, because the brain is changing all the time. And you can say, well, is it really changing because you seem like the same person? No, we are going to leave out introspection. As you just saw, you can't feel spikes. So if you can't feel spikes, you can't introspect about it. Then, once you've acquired this data, you need a suite of tools for circuit manipulation. Because once you measure it, you want to control it. How do you control this activity of 10 micrometer neuron sitting in a zoo of neurons of different types? It's a zoo, not just a crowd. Then we want to link all that activity patterns to behavior so that we start getting from the brain to mind. Well, how do you do that? The way to do that is to integrate theory, modeling, statistics, and computation with experimentation. So all of these have to work together and we have to make sense of it. How are you going to do that? We'll look at that, but that's a mandate. The good news is at the end, this sounds impossible but doable. Then after doing all this, which can be done only in animal models, you start working towards technologies to figure out what's being measured in humans. Because right now, the best thing we can do is MRI. And MRI looks at the activity of about 1 millimeter blob of brain in which you have millions of neurons over a period of 1 second, in which you have thousands of spikes. So you need to look at individual neurons. So we need to figure out how do you relate this MRI, which shows interesting stuff, to these single neuron spikes. Finally, develop technologies to enable collection of human data. We need to start not just have these technologies relate to existing one in this, and of course, disseminate knowledge and training. And I'm going to charge a lot of money for this because they are going to be putting in $100 million to do all this. So we already started with this. So this is going to be $1 million right now. We are going to do the rest of it. And for the purpose of this talk, I'm just going to focus on these little shaded areas, which are the things in which we have done a little bit of work over 10, 15 years, to show you how this could be done in our hands and how you guys can do even better. All right, so do interrupt. So all of this excitement that we are going to be talking about started with this patient named HM, who died actually last year. But this started long ago. Around 1957, this guy HM had a problem. He had unfortunately very bad seizures. And all kinds of drugs they gave him, they could not be controlled, and they had to take out a certain part of his brain. And that part of the brain is called the hippocampus. We are going to use that word many times, so I'm going to throw that word at you. So that's what happened. They had to take out that part of the brain. The seizures indeed stopped. But he had a strange problem that came up after that. And that's well known, happens many, many times, that after the surgery, HM could not learn facts or events, such as if he were to sit in this talk, he goes out and comes back, he will not remember anything within 2 minutes. But he could learn to ride a bicycle. Not only that, every single thing, pretty much, that he had learned before the surgery, he remembered. And a really bad joke is that he knew the name of his surgeon. But that's the last thing he learned. Then it was gone. But, so that was the disease, it was sad, and this had to happen. And people were puzzled. So there were 2 lines of research that started at that point. One was to figure out how to do better surgeries, how to cure epilepsy, which is still going on. And why is it that this weird disorder came about when you lesion hippocampus? So that kind of diverged a little bit. People spent a lot of time figuring out what's going on. A long, long time went by. This is 1957. And all the way in 1973, when a bunch of basic scientists were fooling around with a rat brain, they discovered something completely unrelated to this, seemingly, which doesn't seem to have anything to do with epilepsy or learning facts and events, which is just a rat walking around. And that was this. Why did this happen? A major breakthrough occurred at that time. In 1973, in London by these two guys. He was an electrical engineer, biologist, and I'm going to show you a video and generated by these very bright people in our lab, Lavanya, Zara, and Jason. So I'm going to get out of this, and we are going to listen to actually a single neuron's activity. So I'm going to shut up. What you're going to see is a rat walking by. His movement in space is shown by a blue dot. His position is shown by a tiny blue dot, and it just spikes when the neuron fires. He's doing random stuff in the maze about meter by meter. Just boom, boom are individual spikes. Notice he's doing all kinds of stuff. He's walking all over the place. There's bits of chocolate sprinkles falling all over, so he likes the chocolate rain. He's picking up chocolate all over the place. That's why he's running around. So his behavior is all kinds. He stops, he eats chocolate, he runs. And notice that no matter what he does, no matter what angle he approaches from, and so on and so forth, this one single neuron, this is the activity of one single neuron in hippocampus, that neuron seems to be active only when he is in one particular place, nowhere else. So something changed fundamentally at that time, because people were thinking of this disease, people were thinking of curing epilepsy, something was wrong with the circuit, must be related to learning and memory. Instead of that, there's just a rat walking around randomly, seemingly doing no task at all, just picking up pieces of chocolate. And you find neurons which have map of space. Fascinating. So that question becomes really, really funny. What is that space? And that was my obsession during my PhD thesis, structure of physical space-time. But let's imagine how deep this is. So if I were, let's do this thought experiment. I'm going to say, here is this arbitrary spot right here. I'm pointing to it. And if I were to ask any one of you to come here, I'm moving away from here, and say point to that space, you're going to be able to do it. But you cannot feel that space, that point. You cannot touch it. You cannot smell it. You can't even see it. And still you can do that. And we all are doing this, and that's what this rat is doing. He's just walking around randomly. And there are neurons which seem to code for that abstract thing called space. It is not just a response to a stimulus which is important, such as an oriented bar, very important, or a smell or a reward. It's just seemingly an abstract piece of space. And the brain is making a map of it. All animals must do it, because by definition, animals are animate. And when animals are moving around, they better know where they're going, or they're going to bump into stuff. They're going to get hurt. So animals must make maps of space on the fly, in one trial. You don't have multiple trials, or you're going to bump into stuff. And make a map of abstract space which is not only accurate, which is not only made on one trial, but that's a map that everybody has to agree with each other. Because if your map of space did not agree with the map of space of a tiger, you got bad news coming. So how does that work? So first, this recording technique. I showed you this beautiful activity that these people in the lab recorded. How did we do that? So let's get into a little bit in terms of technology. How do you pick up the activity of one neuron that's 10 micrometer in diameter while the rat is running around and doing all kinds of stuff? So the idea is neurons are really close packed. There are two neurons, A and B, and they are so close to each other, they fire spikes, and all neurons fire spikes which look more or less the same. You can't tell them apart, so what you do is that instead of putting one electrode, you put a bundle of 4 electrodes. The tip diameter is 15 micrometer on each of them, and this is a bad drawing. They are twisted together, but that's an idea. There's a bit of gold and a bunch of nanoparticles at the top, and what happens is that this neuron's activity will be larger on this electrode but smaller on this electrode, because 1/r² dependence on electrical field. And this neuron's activity will be larger on this electrode and smaller on that one, so that if you looked at the amplitude of spikes from both of these neurons on channel number 2 or channel number 3, they will look more or less the same. But if you looked on channel number 1, which is this one, then this will be larger and that will be smaller. And on this one, you will have the opposite. So you can separate two neurons like this. You can make a plot now. You can look at channel number 2 versus 3. There's a cloud which is the same. You can now look at 2 like this, channel number 1 versus number 4. They separate out. And after a lot of effort, which people in the lab put in, what you can do is you can build this circuit. Another bright student who just graduated from physics, Daniel Aroni, he built a device that we can put in electrodes, not just one tetrode in the brain, but 24 tetrodes at the same time, and target each electrode to different part of the brain. So you can pick up multiple types of neurons from multiple parts of the brain all at the same time chronically while the rat is running around and doing his thing. Those are the devices. I'll not get into this. Here's the result when all this works well, which does work very well. This is the data I recorded, but there are much better data recorded in the lab these days, where you get pictures like this, where x-axis is the amplitude in millivolt. This is about 1 millivolt. Y-axis is the amplitude in another millivolt, channel number 2 versus 3. And you can see this blob of cloud where this blue neuron and yellow neuron look the same. They separate out another combination. This is 2 versus 4, 3 versus 4, so on and so forth. You can, by doing this triangulation across these different channels, you can pick up activity of a lot of neurons which are right next to each other. Because they are right next to each other, they are more likely to be connected to each other, which means they'll have synapses, which means that they are going to do something to each other and we can figure it out because they are connected. So that's the power of this. This works day number 1. This is day number 33. Our record right now is up to 9 months. We can record activity of neurons, which is a pretty long time in a rat's life. Becomes a young man and becomes an old man by the time. They live about 2 years in a while. This is the new development we are all working on with several companies. What we want to do is that we want to make this device even more precise. The device resolution is 200 micrometer. But we know neuron size is 10 micrometer. So instead of having screws and screwdrivers with which we move this, we want to replace those with motors. This from top to bottom is just 4 centimeters. We can put 25 motors, put them in servo control and adaptive behavior of these circuit. We can move them with a resolution of 1 micrometer, and we can sneak in close to many, many neurons at the same time. We can talk about this some other time. I want to move on. But the bottom line summary for this part is this multi-electrode technique. You need engineering tricks and abilities such as microcontrollers and motors. You need adaptive control for that. We need alternate materials for the electrodes itself. Right now we are using platinum iridium or nichrome. There may be other materials including nanowires which can get into the brain without causing much damage. We need new kind of electronics which is low noise. Because the amplitude of the signal, even though it's 100 millivolt at the source of the neuron, if you're right next to the neuron, it's going to die because the electrode is going to poke and it's going to be dead. If you want to be slightly far away from the neuron, signal decreases, the neuron is happy, but now the signal is small. So we need low noise amplifiers and A to D converters to do that. Once you go far away from neuron, you get hundreds of neurons activity at the same time. Now we need to separate those using fancy algorithms, which there is a rich tradition in computer science in signal source segregation, independent components, PCA and so on. You need this technique to be scalable. Right now we can go to 25 tetrodes, we can record more than 100 neurons, we can stick in more electrodes. Looks like there are a lot of electrodes, but the amount of damage they cause is minimal because each electrode is only 15 micrometer. Even with 100 electrodes in a 1 millimeter area of brain, you're causing less than 1% damage. So it's reasonably non-invasive. This allows you to measure activity of multiple brain regions at the same time. You can look at one circuit, look at another circuit, how they're talking to each other. We're going to look into that. We can reach deep into the brain, not just at the surface. In humans, it is applicable too. You can do these chronic and long-term measurements, and you can even stimulate the brain very precisely from the tips of these electrodes. So those are the things which are possible here. Now let's move on to these place cells. So can we use this to get further into this brain initiatives? So there are some place cells. How do we understand them? How do we make sense? Where did they come from? How do you go from place cells to finding your way in the world? And how do you learn space? So the first thing we do is to simplify the problem. That's what we do in quantitative sciences and biology as well. So forget this two-dimensional complicated navigation, finding your way, one-dimensional problem. And I'm going to show you that even that one-dimensional problem is something we take for granted, but every single moment of your life, you're solving that problem without knowing it. So let's start with that. Let's imagine I was close enough to you, and I moved my arm like this, at this speed, okay, 1 meter per second. And I asked you to catch it. If I was close enough, anybody can do this. Now let's imagine how the brain works. The way the brain is supposed to work is that information comes from the retina, right there in the eyes, goes through 17 different processing stages in the visual cortex, then it goes to the motor cortex, you generate a command to lift your arm, goes to the spinal cord, and then you touch something. Well, it turns out, much as how great the brain is, it is a little slow. Especially a person like me, really slow. It takes 1/4 of a second, even if you are the fastest monkey who is really motivated. 1/4 of a second. That's the amount of time lag between a simple thing. Just catch this and you get a reward. If you were doing this, if you were perceiving this bar of light and touching it, you will never do this, because in 1/4 of a second, this has moved 25 centimeters, and you'll be constantly doing this. You don't do that. And in fact, if you look at babies, that's what they do. Babies can't catch this, while we can play tennis. So constantly you think that according to the way the brain is operating, you're responding to stimuli. But no, actually. Your brain is taking up a little bit of chunk of information from the world, and it must, even to do this trivial thing, it must immediately generate a perception of what must be coming up next. And next is just 1/4 of a second, at least. All animals have to do this. And there are beautiful illusions people have created to show that this is happening. I can bring that. For example, if the same bar of light was moving in front of you, and while it is moving, if I were to flash at this point, if I were to ask you where did it flash, you will say it flashed here in the future rather than where it flashed. The faster the bar of light moved, you will say further out in the future it flashed. So we are getting tricked by this. We are constantly living in the past and predicting the future every single moment of your life. And when you're walking and so on, it's devastating. You're going to fall. Imagine the number of things that can go wrong with this simple thing, and how it can manifest in complicated things and all kinds of diseases we can't even imagine. So let's imagine that simple problem, which is absolutely crucial for every animal. How can we solve this in this crazy brain synapse, quadrillion synapse circuit, and start making sense? So we're going to make a one-dimensional problem of learning causality. That's even a binary thing. What is cause and effect beyond one dimension? Just binary. So time, fortunately, goes in one dimension. So that is time. Let's now imagine a stimulus that goes by, went by like that. That's a bar of light you're going to touch. Let's imagine there were 2 neurons in your brain. Neuron number 1 and neuron number 2. Let's imagine each neuron has a place, field of sorts, some bump of activity somewhere. I just showed you these neurons do that. And let's imagine the y-axis, this is roughly the probability of spiking, because neurons is firing reliably but probabilistically, not perfectly. This circuit will be useless no matter how many neurons there are, because everything is simply telling you where you are or what is in front of you. It is not going to tell you what is going to come next. And if you don't know what's going to come to next, you are in really bad trouble. If we can do one simple thing in this circuit, very simple, which is that if we can grow a synapse from this neuron onto this neuron and have that synapse excitatory, then what's going to happen after this learning, going from left to right many, many times, is that when this neuron is activated, that neuron will inject some current into that neuron, and as a result, this neuron will pull that neuron's activity earlier. It will generate a hallucination or percept of what's going to come next. And then this relay will work. You can chain many, many more neurons, and these neurons with learning that this followed by that will learn that. So question is, how does it work in real life? This is a physicist's dream. Very simple ideas. Can it actually work? Well, it turns out a very, very famous psychologist named Donald Hebb proposed in 1949 this idea, which I have simplified even beyond. He wrote a book called Organization of Behavior. They're very famous for this. He proposed the following idea, that if a neuron A repeatedly activates a neuron B, the synapse from A to B becomes stronger. And if this rule could be implemented in the brain somehow, then everything will work out. Of course, he was a pioneer. This was the first step. So he couldn't tell you how this will be implemented in the brain. What kind of synapses and circuits will do that? How will they do that? And so on. Many people, and nor could he figure out how that will result in behavior. One synapse versus quadrillion synapses. How? Where is that memory out of synapse becoming bigger or smaller versus pattern? So many bright scientists across the world worked on this. Several of them here, like Kelsey Martin here, who figured out these synapses properties. They do amazing things. And turns out there is one kind of synapse called NMDA receptor dependent synapse which has exactly this property. But it comes at a price. It has that property. It cares for that causality, but it cares for that causality only for 20 milliseconds. So now you see a problem. We wanted to solve a problem of how to predict things on a time scale of 1/4 of a second, 250 milliseconds. We find a signature called a synapse which can do that, but it's even faster. If neuron A is activated before neuron B, only within 10 milliseconds this synapse learns. If it happens 250 milliseconds, nothing happens. So something funny, additional stuff, mystery. We are going to get into that a little bit. How is the activity of many synapses coordinated during behavior, and how does that result in perception, cognition, behavior, and so on? So we're going to do that one-dimensional experiment. Instead of the rat wandering around in 2 dimensions, the rat's going to wander around on a 1-dimensional track, because we want to see, will the place cells become anticipatory? So that's a track. Instead of chocolate rain all over the place, now it's a little harder task. The rat gets a small piece of chocolate here. He has to run all the way to the other end, and then he gets a piece of chocolate. If he goes halfway and turns back, he doesn't get it. Pretty binary, 1-dimensional task. These white dots are the activities, the position of the rat at every given millisecond, resolution of 30 milliseconds, spatial resolution of 0.6 centimeters, and every colored dot is the spike from 2 different neurons. Blue spikes from one neuron, pink spike from other neuron, and so on and so forth. These are just 2 out of hundreds of thousands of neurons in this part of the brain. Can they learn this, and how will they learn this? So I will not get into this theory too much in detail, but I'll just tell you briefly that if you simply took a very simple circuit called a perceptron, which we study in engineering quite often, which is one neuron gets input from a bunch of other neurons, very simple circuit. And if you put this Hebbian synaptic plasticity into this circuit using NMDA receptors and 10 millisecond timing and so on, if this was the connectivity pattern before, when the rat's going from left to right, with learning going repeatedly from left to right many times, this circuit will shift its selectivity. This synapse will become strong, these will become weak, and the whole thing will shift back. That's what this simple theory predicted. And then we went back and checked in the experiment. That's what happens. So this is now not theory, this is an experiment. This is how the rat can learn to predict every single step, what is going to come. These blue dots, blue lines are the spikes from same neuron on trial number 1 on a given day. The rats simply walk back and forth. And blue spikes are the spikes from the same neuron just 5 trials later, within 5 minutes, really fast. Rat goes from left to right, first spike occurs early, lasts somewhat early, peak firing rate goes up, and this entire shape becomes asymmetric, which we believe is a footprint of that asymmetric Hebbian plasticity. And this can allow the rat to predict a relay race between these hundreds of thousands of neurons. Every neuron in the beginning, when you come to this room for the first time, is going to tell you where you are. And as soon as you do it once or twice, all the neurons are quickly telling you what's going to come next, based on what you're doing or based on what somebody else is doing. And that gives you how that causality, which is a basic thing as far as the mind is concerned, or the temporal sequences or movement path that we in behavior perceive, how can they come about from simple circuit ideas, from synapses to circuit and behavior. So there are many properties of these, and there's a surprise at the end. So let me summarize, and that will tell me how to go to the next step. The place cells double their firing rate within 2 minutes, 2 to 5 minutes, double. It's not a small amount. They become asymmetric, acquire this shape. They become more anticipatory. Neurons start to anticipate by about 20 centimeters what's coming next. This depends on the receptor and MDA. If you block the receptor, it doesn't happen. Shows you that's what is the likely reason. This is consistent with many of these models. This is supposed to be the emergent property of Hebbian synaptic plasticity. That perception or learning of causality is supposed to be one of those little links. Similar ideas, now that they are general, perceptron can be applied to any part of the brain. Several other labs are seen in many, many parts, many species, rats and xenopus or frogs and primates and cat visual cortex and so on. And these results last at least for half an hour. So we were all very happy. We wrote lots of papers and so on. But then, while I was looking at what was going on, something funny happened. Turns out all these massive changes which are going on in place cells, most of these changes seem to be gone after a period of sleep. So what is that? Why is it going away? This very precious thing that I tried to convince you is so important that without which you are going to run into things or get eaten up. That causal learning seems to be going away. How can that be? Why is that? Or is this relevant at all? So story gets more complex. Turns out one thing which is crucial for this, which again is the stuff that you cannot perceive, is the brain rhythm. So if you looked inside the hippocampus, apart from place cells, you looked at the activity of many neurons. Not only is each neuron firing at a different place, collectively they are also firing in a rhythm. So the x-axis is time in seconds. This is half a second from here to there. From here to there is 1 second. Red is the velocity of the rat or speed. And in green is the average activity of ensemble of neurons called EEG or local field potential. And you can see it is a systematic fluctuations which are noisy at about 8 hertz. And turns out that this 8 hertz rhythm that brain is generating on its own. 8 hertz is something you can perceive. It's about that. That's what's going on in your brain as soon as you walk. As soon as you stop, that rhythm goes away. You fall asleep, the rhythm is gone. As soon as you go into REM sleep, where you start to dream, the rhythm comes back. Happens in humans, happens in rats, happens in all species. So something funny comes up now. There is a rhythm that brain is generating as soon as you explore space, which you can't feel, but it is there. It's somehow important for all kinds of things that you are doing. Turns out if you mess with this rhythm, leave the circuit intact, pharmacologically, you mess with this rhythm, you have serious problems in variety of disorders ranging from schizophrenia to learning and memory. Another example of how the basic stuff that's going on has strange ramifications, things which are seemingly unrelated to each other. And I'm going to skip how this works, how this rhythm enables this time compression and generates a digital code from analog, and I'm just going to move on to the next thing. Any questions so far? I'm going to jump through the next thing. Yeah?
Does that rhythm affect the notion of time? Is that why we all have at least a somewhat consistent notion of time?
It's a very good question. And papers which have started to look at it within the last 1 or 2 years have found that if you ask the subject to keep track of time, these rhythms appear once again. And there's an idea that these rhythms may be playing the same cells which are keeping track of place are also keeping track of time. This is very recent, just last 1 or 2 years. It's fascinating. Anything else? Okay. So in this so far we said, well, somehow there's a neuron that's responding somewhere, and as long as we take that as God-given fact, the rest is going to work out, it's going to learn sequences and so on. But the question that we did not answer is, what is that space? Why is it that there's a neuron that spiked here? You saw that big arena, it was spiking only in restricted area, nowhere else. That abstract space, the thought experiment that we did, what is it that brain is putting together? If I were to ask you, how did you come and touch that region of space? You will say, well, I remember you were at some distance from the screen, some distance from the podium, some distance and so on. I put it all together. So how does the brain put all these multiple modalities together and create a representation of something that doesn't exist? How does it do that? Well, in order to do that, we had to jump into novel techniques called virtual reality, and these very, very bright people in the lab developed this, And the idea is this, that when you are walking around in the real world, everything is changing at the same time. You think that when you are walking, only the visual scene is changing, but the smells are changing, the sound of the AC from here is changing, the airflow is changing, the smells are changing, the texture on the ground is changing, and if I didn't keep track of it, I'll be tripping all the time. So not a good idea. So how does the brain actually put together space from many modalities? So we thought, let's simplify this. We want to control that visual modality. So this is a virtual reality apparatus we developed. That's our favorite guy rat. It's non-invasive. We just put a little vest on the rat's body. These are very, respectable people. They wear a vest and they're butt naked otherwise. There it is. Very nice. And they have a little, unknown to them, we put a bit of Velcro at the back. And that Velcro holds them. It's a vest that comes with a price. And right there is this Velcro. They still tolerate this, and they're on this giant floating ball made from styrofoam that's floating on air from the bottom. There are a bunch of cool techniques here. These guys can tell you how it works. And as he starts to walk, this ball starts to rotate. As the ball rotates, there are these microcontrollers here that pick up the movement of the ball through this computer mice. So rat walks on a big computer mouse, and other computer mice pick it up. Many jokes there. And those mice, through microcontrollers, send the information to a computer, which sends the information to a projector that's kept here, that sends the information to this mirror, and that sends an image all around him. And now, through this complicated set of things, we have intervened between the rat and the world around him. We have complete control over it. When he walks one step, we decide how far the world goes, in which direction and which part of the world goes where. So this is how it works. This is very dim, intentionally so, because rats actually get stressed out in bright lights. Cameras don't work well, so some experimenter is going to move this scene. And you're going to see, even though the camera is outside here, the rat is inside, this gets covered up, of course. It's all in a big sound-insulated, light-insulated room. So he's on his own, so the perception is pretty compelling. But even from the outside of this crummy light, you will see that this is reasonably compelling. So you will see this big pillar coming towards you and going over your head, but of course there is no pillar there. And the rats buy into this all the time. In fact, we have tons of videos to show that the rat goes there. There are no rewards for the pillar because they're above. And when the pillar approaches, they stand up and try to touch it with the snout. So unrewarded, but they buy into this. So it's a pretty compelling thing. And now what we are going to do is to show you how quiet this system is, because we want to make sure that the space that he's running on is defined only based on the visual scene. And we are not going to mess with the visual scene. We are not going to put them in black hole, wormhole, teleport, all those cool things we can do. But we have to be very disciplined in the first part and figure out that very simple thing, what happens when you just walk from here to there. And you will see in a couple of slides actually very surprising set of things happened. So we're going to turn off all the sounds in the room. We are going to listen to the rat's footsteps for the first time as he's running. So that's what happens now. We're going to do that. It's really loud. So that's a task he's doing. He's walking on that same linear track that I showed you before. His job is to go from back to back. He goes to the other end, and right in front of him is a little tube from which sugared water comes in. And what he has to do is to go all the way to the other end, and when he reaches the other end, just like in the real world, he got a little bit of a reward. He gets a bit of sugared water, and he really likes it. And while he's drinking that, either we ask him to turn the world around, or we tele-rotate the world for him so that we minimize the variability in his behavior, so he's just walking straight. And he does many, many interesting things that we did not ask him to do. We only asked him to go all the way to the end and stop. What he does is that he every now and then falls asleep, like most of us in these boring seminars. I don't care. But when he falls asleep only at the end of the virtual world. Even though it's equally comfortable everywhere, even though there are the same smells and sounds, he sleeps only at the end, invariably. So they really buy into this. So what we are going to do now, this is something we didn't plan on. We have new research avenues. Now what we are going to do is we are going to turn off the footsteps of the sound. We are going to listen to one neuron's activity. Remember, these neurons are supposed to be place cells, and they are supposed to spike at one place. That was a big discovery in 1973. No matter what you do, the neuron fires at the same place. As you can imagine, that doesn't happen, but in an interesting way. So, oops. So we looked at these neurons. So these are many, many neurons. So we compared. First of all, you build some complicated apparatus. All kinds of stuff can happen. So what are these neurons doing? So these guys, these authors, Pascal, Revasard, and Ashley Keys, Bernard Willers, and many others, they really, really worked hard. Made all these biology and surgeries and behavior and EFIS and math, all of it work. And they recorded more than 2,000 neurons, which is a lot of hard work. But that gave us really solid foundation to say this is real. You'll see why. So as I told you, with this tetrode, you can pick up many, many cells on a single electrode. Like that, these are all different ellipses are different cells. I won't tell you how. With the same electrode, RW stands for real world. You look at the same electrode in virtual reality on the same visual virtual maze. Half of the cells just shut down. All these ellipses are gone. In fact, across the ensemble of these 2,000 odd cells, if you find on any given day on a given track in the real world, as it is known, not all cells in the hippocampus are active. Only about 40% of cells are active in any given maze. Other 60% are active in other mazes. They are completely silent pretty much. In real world, 40% of cells are active. In virtual reality, only 20%. So about 60% reduction in the number of cells which are active just because the rat is running in a virtual world. As far as we can tell, based on his behavior, he's doing as well as in both worlds. He seems as happy, he drinks as well, he sleeps as well. Only half as many cells are active. But the cells which are active have the same level of activity, the same firing rate. This is the firing rate of cells in hertz. They're spiking away at more or less 3 spikes per second in both cases. But half of them are just not there. And funny thing is that the same thing happens when you look at the track versus goal, but I'm going to run out of time, so I'll just skip through that. So then we're going to listen to this one single cell and look at these cells which are active. How are they making space? Which combination of visual cues is driving them? Can we make sense? So let's listen to this cell. Okay, so one cell's activity. So notice this cell spiked at the end of the track. So you walk from here to there, cell spiked more or less at the end. Now world is turned around, starts to go the other way. If it was a place cell, it should spike right there. Nothing. It spikes at the other end. And this happens routinely. So again, now he's at the other end. The cell just spiked when he was coming from here to there. Here, it's not going to spike. He goes all the other end, and there is the example of a nap he took, a little nap on the place. In the next part, I'm going to tell you what good those naps are. Very crucial for actually perceiving space. But we're going to go through that. So this goes on all the time. So these cells, very reliably, in virtual reality, instead of encoding absolute position, as they were doing in real world, they are actually keeping track of distances. And I'll show you some statistics to show this is very reliable. But basically what happens is that we found some kind of relativity in the brain, that just like physical space is supposed to be absolute, now we think it's relative in the brain too. When one modality is isolated, these neurons do that. This is in more pictorial form. This is in the virtual reality. Here is a rat walking from left to right. The track is 2.2 meters long or 220 centimeters. Neuron didn't fire here at all or very little, and then fired a lot, the asymmetric place field. More or less at 140 centimeters, the rat fire, the neuron spikes when he goes from left to right. When he goes from right to left, the neuron does not fire at 140 centimeter, instead spikes at 70 centimeters. Whereas in real world, these are still the place cells. It's not like we ended up in a weird part of the brain. Identical visual tract. Here's a neuron in the real world. Same neurons we find, they do that. This neuron spikes more or less at 50 centimeters when he goes from left to right. does not fire like this one on the other side, but fires exactly the same position. So these neurons are doing something very different. It's telling us something about that space that we take for granted all the time. As long as we are moving, we are generating a percept of space. We think you are making that space based on visual cues. That's what you would have told me if I had to ask you how you found that little spot in this room. But your brain is doing something very different. Yes?
You get the same effect if the rat has to turn around.
Wonderful question. Yes. We taught one rat to do both tasks. So either walk back and forth when it's flipped around, or he has to turn the world around himself. Exactly the same effect. Yes? Anything else? Yeah.
So then in virtual reality, there's a lower level of activity because there's less input.
Yes.
Would the reverse be true, or is there some upper bound on how much activity your brain can handle or something?
We don't know. I mean, reality, we think that's the reason. We think that when you are making perception of space, you need multiple modalities, such as sound and smells and so on. And we're actually building this new version of virtual reality, where instead of just the light at every position, there are also sounds at every position, and smells as well. And we think that when you put all that in, you will approach the real reality. And then the brain will be fully active, but then the question is, how are they all put together? Which is a big issue. Okay? And that's what happens there. So in this complicated plot, I want to show you that lots of neurons do that. So what we are going to do is compute so-called correlation matrix, because in the brain, as you know, it's all about the matrix. So at every position, you have lots of neurons active. So you look at the pattern of activity of all the neurons rather than the activity pattern of one neuron. You look at the pattern of activity of all neurons at one position, take that vector called population vector, look at how similar that vector is to itself or not at different positions. Compute roughly the dot product if you wish. That's shown here. And what you see here is this is the forward running direction in the real world. This is the backward running direction, and the color indicates how correlated it was, close to 0.1, close to unity. And you see that all neurons are active at the same position. In the forward direction, if it spikes at 110, in the reverse direction fires at 40, so on and so forth. In virtual world, population vector across all the neuron never does this, does the opposite. It spikes at the same distance. And there are many more statistics. I'm going to jump through this. Back to somebody's question, turns out the brain's rhythm, which could be keeping track of time, actually slows down. Interesting story. We are not going to get into that. So going forward, we want to do many more things. For example, instead of the now, we have established the baseline sort of a thing, found many surprises. Now we want these guys to do complex tasks rather than just walk back and forth. To figure out where they're going, we want to include other sensory modalities such as smell and sound, have them all in register. It requires new technologies, new controllers, new codes to control these and make sense of this, how to put it together, and measure other aspects of behavior too. So he was just walking back and forth, and we are simply saying how fast he walked and where he stopped. But now that we are in virtual reality, and this little tube is right in front of him, we can also measure his expectancy of reward. Because whenever, like Pavlovian dogs, which salivated the expectancy of reward, these guys will lick the reward tube. And we can now make a map of the reward expectancy map and a map of their walking map. These two are supposed to be very different worlds. One is so-called Pavlovian conditioning world, which is all about reward. Other is about just objective thing called space. We have finished some of this, and that's a paper that just got accepted yesterday. So interesting stuff going on. We can tell you something about it. But I want to wind this up. I know somebody has told me that this goes up to 5:45, but I think that'll be too long for all of you. So I'm going to wind this up in 10 minutes and tell you the last part of this story. One part very quickly is the need for speed. Again, thinking from basic physical sciences helps here. So we agreed that in order to navigate, you need to know how fast you are going as well, where you are going. Where is that information about speed? All we have are place cells. They are coding for where you are. How do you solve that? As we know, in order to know where you're going, you need to know how fast you're going. Where is that signal? Well, basic science tells you that if you want to figure out the signal for speed, it must be independent of signal for position. Why is that? Because at the same place, I can go fast or I can go slow. So the brain must represent speed using completely different measures than the signal it uses to encode where you are. Otherwise, things will get confusing. But you have only one neuron, or an ensemble of neurons, and all of them are encoding place or distance. How can they now also encode speed at the same time? We scratched our heads for quite some time, and the answer eventually appeared to be very simple: Fourier transform. There is this basic theorem in Fourier transform which says that you can represent any signal by its Fourier components, and all Fourier components are orthogonal or independent. So what if we took this crazy idea from mathematics and applied it to brain signal and said, let's just take this ensemble activity, let's take its Fourier transform. We'll figure out how the brain does that. How can brain do Fourier transform? Well, we do cognitively, but how will neurons do is another story. Let's look at it. So we took Fourier transform of this signal, and lo and behold, we found not only a Fourier transform based code, but it used all aspects of Fourier transform. The amplitude modulated, frequency modulated, and phase modulated code for speed. That's how important speed is. And as you can imagine, it is. Because speed can kill. You can get hit. Whereas if you don't know where you are, you can stay put. That's at least what we think. Here is a simple idea. Here is the speed of the rat. He runs, he stops and drinks water or eats food, runs again, that's the speed. Here is the Fourier transform of a signal from his brain in two different bands. This is about 20 to 40 hertz. This is 40 to 60 hertz. And if you can't see this, if you simply look at the amplitude of this signal versus the speed, it increases by a pretty large amount, even though the speeds in these lab conditions are very tiny, only 40 centimeters per second. And these two different bands, they increase the speed very differently. One is a linear one, other is a log. Very simple differential equation to solve. dA by ds is equal to constant versus 1 upon s. This is the phase code. You look at the speed versus the phase of that rhythm that I told you, 8 times a second. You have a signal for speed even in that. At different speeds, you have a different phase. And here is an FM code. For the first time that anybody found an FM code in the brain, a frequency modulated code. As the rat speed increases, the frequency of the brain rhythm itself changes from moment to moment, all the way from 30 hertz to 250 hertz. That's a complicated picture. So the brain, by applying these mathematical ideas, we find a whole new set of codes, very fascinating mathematically and of practical use. And I'm going to skip this dendrite business because it's going to take me way too long. So, and I promise that I'm not going to go on till 5:45. So here's that last part, which is part of this brain initiative, is you want to look at not just one large circuit, but you want to look at multiple large circuits at the same time. So the question is now, well, you can stick some electrodes in two different parts of the brain. How do you figure out how those parts are talking to each other? Another interesting question along with this at the same time is what goes on in your brain when you sleep? Because you lose muscle tone when you go to sleep. But interesting fact that has been known for the longest time is that brain is pretty active during sleep. Only about 10 to 20% less firing rates of neurons overall during sleep than when you are really attentive. So why is the brain active all the time during sleep? What is it doing? One clear answer emerges from introspection, although we are going to be cautious about it, is that if you are sleep deprived, you are pretty messed up. So something to do with learning and memory goes on, but it could simply be because your gut is messed up, your legs are aching. But many, many careful studies have shown that actually sleep has something to do with learning and memory, in particular with memory sequences that you learn, temporal sequences such as playing tennis or such as learning some sequences. Somehow what happens in sleep plays a role there. Has been the longest standing mystery. Let's look at it very quickly. So there are these two giant circuits. One is the circuit right underneath your skull called the neocortex. Other is a circuit underneath it called the hippocampus. And from those studies of this patient HM, when they found this funny disorder he developed, that when he took out his hippocampus, he could not learn new things, but things that he had learned before were present. People came up with an idea that maybe the way our brain operates is that this part of the hippocampus, brain called hippocampus, which has maps of space and so on, that's like the RAM in a machine, in a computer. It stores information for a short period, or maybe your scratch pad right now where you may be jotting down notes. And then when you go to sleep, that's when there is no sensory information coming in, there's nothing to learn from outside. That's when the hippocampus dumps all that information to neocortex, takes a backup, sort of. And that was the idea proposed by a bright man named David Marr way back. So maybe these two regions, that's why the brain is active. That during sleep it is dumping information, and that's why HM remembered all the information before because it was stored in neocortex, and neocortex was intact. But now the hippocampus is gone, so a scratch pad is missing. And that's why there's nothing to back up. So that's the idea. But it has remained kind of unverified and a major challenge. So sleep has, roughly speaking, two parts. One is the REM sleep, which we all think we are doing, which is so-called where we have dreams. But that's in adults is only about 10% of the time. And as we grow older, we dream less and less. And that has nothing to do with our cognitive abilities. Other part, major part of sleep is so-called slow wave sleep. And if anywhere to ask you what do you do, that will be your most accurate answer. It's not you're doing engineering or physics or biology. It is slow wave sleep. Because that's the most common thing your brain does. As soon as you sit down, you stop doing anything, you relax a little bit, your entire neocortex more or less synchronously develops another rhythm. Pretty synchronous, about once a second. Unlike that 8 Hertz rhythm that appears in hippocampus only when you are running, the whole of your neocortex all the neurons start to become active and inactive. In fact, it behaves like a two-state system, not a sinusoidal oscillation, a binary oscillation. Has been well known, and in fact very recent studies done with medical students found that if you simply strap on a bunch of magnets and stimulated the brain at 1 Hertz, you will remember stuff better. Very strange. And you stimulate only during sleep, slow wave sleep, this improvement occurs. If you stimulate during behavior, if you stimulate during REM sleep, there is no improvement. So if you enhance seemingly, this is still early, seemingly enhance this brain's 1 Hertz oscillation, you somehow cognitively remember better. Why? We don't know. But we are going to get into that just a little bit. If you now look at hippocampus, while the whole neocortex is doing this 1 Hertz rhythm, the place cell neurons in hippocampus, they seem to not care for it at all. They show something called large irregular activity, which is exactly what it is. There's a lot of activity, it is large, and it is irregular. So now, what happened? Why are these two things doing these different things? One is going at 1 Hertz, it is synchronous, the other doesn't care. And the two are supposed to talk. And one is supposed to dump information from one to another. They're not even talking, seemingly. So what's going on? So we developed another technique where you not only measure the activity of neurons from far away, but you go very close to neuron and measure its membrane potential itself, rather than just spikes. So in blue is the average activity of a lot of neurons. This is time in seconds, about 7 seconds of data. That's the neuron from which this activity is measured. This is the amplitude of signal in 2 different units. And you can see in red is the membrane potential of this neuron. It shows this binary oscillation. These are called up and down state oscillations. The brain behaves like a two-state system, turns on, turns off. No spikes here, spontaneous spikes here, so on and so forth. Goes on throughout your period of sleep. Now we want to look at this spontaneous activity, these triggers that brain is generating. Look at this pretty sharp transition. What we want to do is to pick up these triggers and say, okay, exactly when this neocortex woke up, what did the rest of the brain do? Because there's this burst of activity. We'll use it to map the functional connectivity of the brain using this trick. Because we can't measure every neuron's activity yet, it will take many years. To do that, we had to develop bunch of algorithms, and again the credit goes to this very bright graduate student who is now a postdoc. And then we focused on a part of the brain which is just between the neocortex and hippocampus. It's a part of the brain called entorhinal cortex, and the entorhinal cortical neurons, they too have place cells, but the place cells are even funnier. So the same experiment that I described to you, when the rat is walking around randomly, entorhinal cortical neurons too have place cells, but those place cells are really, really funny. Those neurons don't just have one place field, but they have multiple place fields. And these place fields are distributed on the vertices of a triangular lattice. Imagine how strange this is. So here's a rat that's wandering around all over. This is a top-down view. And these vertices are drawn for a visual guidance. And the cell spikes either when he's here, or here, or here, 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 and so on. And not in between. And this was discovered very, very recently, just 2005. So that's what the brain is doing on its own, which is again something that if you wanted to fix the brain, you would never think. And many, many people knew how to make theories about the brain for the longest time. People have made robots which navigate in foreign environments based on just place cells. They never used these cells until 2005 because there was no need. Things were working perfectly fine. Robots could easily go do all kinds of things in strange environment, and then people found, well, this entorhinal cortex has these neurons which have this triangular lattice. How does it come about? Why does it come about? What is its use? Fascinating questions, lots of experiments going on in our lab and elsewhere. We have no idea is the bottom line. But there is something really interesting going on which is not something you would have thought as recently as 2005. So we decided, let's investigate these neurons not when the rat is running, just when he's lying down asleep, in slow wave sleep. Is there anything special about these neurons? And of course there is. Turns out these neurons throughout the period of sleep are behaving like a memory device. So here we go. So here's this part of the brain. It's way back of the brain. That's where the cerebellum is. This neuron is buried in there. We reconstructed this neuron. You measure this neuron's activity, and you see this very nice activity. They are very active, unlike neocortex, which fires one spikes in a couple of seconds. Very robust activity, going up and down and so on. Now, Thomas Hahn and James McFarland did an even more difficult experiment. Not only measured the membrane potential of this neuron, simultaneously they measured the neocortical activity of the two circuits, which is giving input to this, and aligned this activity, and that's what you see. In gray now is the neocortical activity going up and down once every second. And what you see is that as soon as neocortex wakes up, just a little bit after the entorhinal cortex wakes up, but when neocortex goes to sleep, it remains pretty robustly active. 2, 3, and then it comes down. In this case, it lingers a little bit. In this case, it bridges just one cycle. So it's a stochastic memory, so-called persistent activity, which is going on all the time. On the other hand, if you look at very similar neurons, which biochemically look identical, just half a millimeter away in the so-called lateral entorhinal cortex, they too show this up and down activity, but if you looked at the local field potential in neocortex, they are not lingering at all. They are just locked in. Two parts of the brain, just less than half a millimeter apart. They look like the same neurons. One has memory pretty much all the time, the other doesn't care. And these are the neurons which have the triangular lattice kind of activity, and they drive hippocampus. And this is all on an effect. I'll spare you the details. This activity is actually quantized in the units of neocortical activity. It's not just coming out of randomly. So if you looked at the neocortical activity, the duration of this memory-like activity varies anywhere from about 1 second all the way up to 200 seconds actually. It can remember the neocortical activity up to that period in a very long way, but it can either contain integer multiples of neocortical cycle, 1, 2, 3, 4, 5, and so on. So it's pretty strongly driven. These are super long memory activity lasting 200 seconds. Neocortex keeps waking up and going to sleep. This guy just persists all the time. We have some theory about that. If you now went and looked at single neurons in hippocampus while the animals are sleeping, what you find is that the same sequence that you are seeing during behavior, rats going from left to right, it takes him about, in this case, 250 centimeters. It takes him about a couple of seconds to go back and forth. If you looked in one particular part of the hippocampal activity pattern called sharp wave ripples, the same pattern of activity that occurs during behavior is now being replayed in hippocampus during sleep. But the replay is 50 times faster. And turns out that if you mess with this replay during sleep, this is unconscious. This is not during the dreaming sleep. This is while you are totally wiped out. You have no memory of what you had dreams. And at that point, spontaneously hippocampus is generating these sharp wave ripples, and 50 times faster than what happened during behavior, that same sequence is getting replayed. But turns out if you mess with that hippocampus's ability to generate this replay, you don't remember what you learned yesterday. Tiny manipulation. People think that's how this communication between these circuits is working. And since I've run over, and I promise I'm going to finish at 5:30, I'm going to tell you just this very briefly that this is what we think is going on. It is not that during sleep memories are actually being consolidated. The opposite possibility also exists, which is that when you are running and we are doing anything, your problem is that you don't know the future. You don't know that one step that you took is going to take you to something important or something completely useless, which is what happens most of the time. But your brain doesn't know that. You don't know that this step you are going to take is going to lead to reward or punishment or nothing at all. So you got to treat this as really, really important step. Otherwise you can't figure it out. So what the hippocampus does, according to this idea, is it learns everything as if it's the most important thing. That's why hippocampus is so prone to LTP, so prone to having synaptic plasticity, just wants to learn everything. And during the period of sleep, instead of making everything that is learned stronger, what the neocortex through this persistent activity is doing is to figure out which of these things are important, and more importantly, that 99% of the things that happened that day which were actually completely unimportant. Get rid of them. By doing that, you can improve the signal to noise ratio in this very high dimensional space of memories and still improve your memory. And that's why this replay can play a role, but I'm going to skip it. I want to summarize very briefly that we talked about these several steps. These are by no means comprehensive. I have been very bad in not talking about many, many other things others are doing, but that's all I could do. Talking of using this, people who are interested in this, we are starting this new affinity group called Neurophysics and Neuroengineering Affinity Group. It should be online soon enough. Those of you who are interested can send us or others an email. We'll put you on the list. I'm also teaching a course called Neurophysics of the Mind-Brain Problem called Physics 188, which is both graduate student and undergrad course, which teaches pretty much the same thing. And I want to end by thanking this very, very bright sets of people, Daniel, Ziping, David, Zara, Bernard, and Jason, and funding from many of these sources. Thanks so much for your time.
A few questions.
When the rat is in virtual reality, the feeding tube, which it most cares about, is stationary.
That's right.
Doesn't that give the rat a different sense of their location?
Absolutely, it can. But they don't seem to, somehow the neurons don't seem to code for that. Neurons don't code for that. They just seem to code for where he is in virtual space, not based on how far he is from the tube. We looked at that actually. Does it depend on how far he is from the tube? Doesn't seem to matter. But it's an interesting question. And one can have another version of virtual reality where the tube is invisible. That would be good. Absolutely.
One final question? Here.
During sleep, patient has a very dream.
Good question. I don't know the answer. Because you see at the end it's subjective. So I kind of stay away from that.
Thanks very much for the speaker.
Thank you.